And especially when thinking about advancing the model further to a stem cell-based model, now we have the cell line, but we can advance it further to a stem cell-based model, then we really go a step further towards personalized medicine and patient-specific models. And I think that's a great glance into the future of organ-on-chip models in general. My name is Maria Warschinke and I'm a PhD student at the University Hospital in Jena. And my research focuses on the establishment of a C. diff infection on chip. And we also want to further explore potential correlates of protection, such as neutralizing antibodies against the C. difficile toxins or whole bacterium antibodies to see how they interfere with the infection of C. diff or the colonization in the gut. So the first point is that there are not too many in vitro models out there yet. C. difficile is an obligate anaerobe bacterium which is able to form spores, and that makes development of such in vitro models quite challenging. So this is the first point, the development of the model. And the second point is to investigate in those therapeutic options, since treatment options for C. difficile so far are limited. C. difficile infections, they are quite complex and they involve a lot of different pathological processes. And the problem with in vitro models often is that they don't provide too many options in scaling up complexity to the needs we have. So with the organ-on-chip, we really have those options. We can increase the complexity to the amount we need. So the maturity of the cells is increased, for example, over 2D models. And we can include circulating immune cells, such as neutrophils, which is part of the immune pathogenesis of C. diff. And we, with that, can form a really physiologically relevant infection environment for C. diff to be able to mimic the hallmarks of a C. diff infection in vitro. You can see the model here. I'm working in a gut-on-chip, and it's a cell line-based model currently. And on the epithelial side, we have C2BBE1 cells. Those are CACO2 brush border expressing clones of CACO2 cells, and when we apply perfusion, they nicely build those 3D crypton villus-like structures. And on the other side of the membrane, we have the endothelial cells, UVECs, and PBMC-derived macrophages and dendritic cells are part of the model. And both of the chambers are constantly perfused. And this enables us to include, for example, the living obligate anaerobe bacterium on the epithelial side, or neutralizing antibodies or neutrophils on the vascular side, and this enables us to mimic the C. diff infection in vitro. So far we did a lot of preliminary investigations with the purified toxins actually, and I'm in the middle still in establishing the C. diff infection on chip. So I would like to show you now the results that we gained with the purified toxins, since they already show the great potential of our model. So for the C. diff toxins A and B, which are the main virulence factors, we could show, for example, that toxin A is specifically acting on ZO1, leading to a destruction of this tight junction protein, whereas TCDB does not affect ZO1, but rather affects like the whole cell layer, as we can see here with the diminished E-caterin signal. Both toxins were able to lead to a loss of barrier integrity, which can be seen here in the increase of permeability. And they both induce an inflammatory process, but especially when there are immune cells present. And we could also show, and this is one of the advantages, by including circulating immune cells in our model, that the toxins are able to induce the infiltration of the neutrophils. And all of these effects basically could be prevented by using neutralizing antibodies. So the barrier integrity was coming to a level like in the untreated condition, and the cytokine release was prevented, as well as the neutrophil infiltration. And we hope that we can see those results then as well when we do the infection with the living obligate anaerobe bacterium. It definitely was. So there are mainly 2 points to consider when thinking about animal models, for example, and we have a disease where the immune component is quite important. Animal models and humans, they have interspecies differences, which makes it sometimes a bit difficult to transfer results from the animals to the humans in the end. And 2D models often lack the complexity that we need. And organ-on-chip models, they are kind of in between. They give the opportunity to have a high complexity, and we can generate the models to our needs. And we have the immune component present. We can investigate host microbiome interactions. And especially when thinking about advancing the model further to a stem cell-based model, now we have the cell line, but we can advance it further to a stem cell-based model. Then we really go a step further towards personalized medicine and patient-specific models. And I think that's a great glance into the future of organ-on-chip models in general.